guess we are ready, right?
Yes.
Okay, so it's a pleasure for me to introduce Elena Rodríguez Vieitez. Elena is a Galician researcher like me and also a physicist. And she has a lot of experience working in the Alzheimer's field, specifically with tau PET. She was for 10 years in the laboratory from the legendary Agneta Norberg in Karolinska Institute.
Yes.
And 2 years ago, she moved to the field of frontotemporal dementia. So probably what she's about to tell us is very relevant for our future endeavors with FTD. So, Elena, the stage is yours.
Thank you very much. Thank you for the opportunity to give this seminar. It's the first time I'm visiting the Fundación CIEN. And thank you, Jesus, for introducing me to Pascual and to, well, Michelle. We already know each other for quite a long time. So I work in Karolinska Institute as a docent in medical imaging and senior researcher. So in this presentation, I would like to focus on projects that I'm working on in the last year or two. So it's a bit of mix of different things, but it's mostly biomarkers. And I'm happy that you interrupt anytime, make it interactive, or stop me anytime. So the topics I will talk about is MRI-PET imaging and peripheral inflammation. And the aim is to find good prognostic biomarkers in aging and hereditary dementias. So I work, it's called Solna campus, it's the main campus in Karolinska. Just wanted to show you. This is building here is called Aula Medica. It's a big conference hall. And I work next to this Aula Medica is a hospital, and it's the new Karolinska hospital. And I work in Bioclinicum on the top floor. We collaborate with different hospitals in the area. So we have hospitals in Solna, but also in the south side, there is a place called Huddinge. And there is also a Karolinska Institute has a center located next to the Huddinge Hospital. I used to work here when I was working with Agneta Norberg. And when I moved to the new group with Caroline Graf, is when I moved to Solna. We also collaborate when it comes to PET imaging with Uppsala PET Center. As you know, it's like a pioneer place for many PET tracer developments. Good, and I also one slide to show you that we also have data from our own memory clinic in the Karolinska Hospital. So part of our research is based on data from patients in the hospital. So this memory clinic has greater than 700 patients per year. And in colors are the patients that get dementia diagnosed. As you can see, as you know well, the most common dementia is Alzheimer's disease. And but we also have vascular dementia, frontotemporal, Lewy body, and others. And then on this side, about 1/3 of patients get mild cognitive impairment diagnosed, and 24% get subjective cognitive impairments. And these two are very interesting for research because we aim to have early biomarkers, right? So these are very uncertain, let's say, prognosis. And they're good also for in the future for clinical trials. Yes, so so you mean if they first go to the primary care? Yes, because this is a tertiary clinic, so they get referred when, yes, yes. So Agneta Norberg that I was working with, she works 1 day a week in the memory clinic, seeing people with memory disorders. So I think one thing I heard in Sweden is that dementias, and so memory disorders, are seen by geriatricians. And others, for example, Parkinson, are seen by neurologists. So I think in Spain it's different because here is neurologists, right? Yes, but there is, I don't know why the reasons is like this, but okay. Yeah.
Things that you have a very early stage population. So
right. Yeah. Uh-huh. Yeah. So already. So this could mean that here they get referred late. and it could be because of maybe primary care doctors should be more trained or to detect or more sensitive tests. This is a good question, but I'm not sure. Yeah. Okay, so but my background is physics and engineering, but I became very interested after my PhD to study neuroscience, and I focus on data analysis and developing biomarkers. So of course, the big biomarkers in Alzheimer's disease are amyloid and tau. But so I've been working a lot with amyloid PET and tau PET data in the first years of my postdoc. But I also understand that these are quite expensive techniques. And also, not only there is amyloid and tau, but even when I was working with Professor Norberg, one of her big interests is the glial cells. So my first project was to image astrocytes with deprenyl PETs. So today I will not talk. I will not focus so much on deprenyl PET because in the last few years I am not working with deprenyl, but we still have deprenyl PET data, and people from the group of Agneta are also acquiring deprenyl PET. So this is still an active research field, even in other dementias, not just Alzheimer's, because gliosis is thought to be a phenomenon, a biological phenomenon that is common to many dementias, to many types of neurodegenerative diseases. Okay, so as in the recent year, I have joined with AMI-PAD projects. As you know, some of you have been involved in AMI-PAD. You were involved, or yeah. It was the end of the EPAD? Yes, yes. Yes, so part of my postdoc in the last, I already finished this project, but has been on data coming from the AMI-PAD, is amyloid imaging to prevent Alzheimer's disease. And this project is very nice because it is from many different centers in Europe, and the aim is to collect amyloid PET and CSF amyloid data. There are many cohorts, and some of them are enriched by recruiting participants with affected family members, like in Barcelona Beta, for example. So you have one of the, this is a very good types of cohort to investigate risk factors. And in my project, I was supervising Arianna Sala, a PhD student from Milan, and we focus on investigating what kind of information we are getting from CSF compared to PET amyloid. And the hypothesis is that CSF and PET are not totally giving us the same type of information, but that we would get complementary information. So we published this paper, and in this case, we use ADNI data. We collected all the largest possible set that had all amyloid PET and CSF at baseline and at 2-year follow-up. And this gave us a sample of almost 900 individuals and out of them at baseline, 300 were follow-up 2 years later. Okay, so basically from previous studies, it has been published already that there is always a discordance between CSF and PET that can be up to 20% of the cases. And this discordance is especially big at early stages in preclinical Alzheimer's disease. Okay, so we follow them up in groups of PET based on dichotomous biomarkers. And we found that when you have a group that are negative for PET and negative for CSF, longitudinally, they don't progress directly to PET positivity and CSF positivity, but they can follow 2 alternative pathways. In one of them, they become first positive in the CSF and positive in the CSF and negative in PET, or the other way. And so the outcome of this study was that these 2 pathways had significantly different factors, such as, we found it interesting, a lower frequency of APOE4/4 carriers when you have PET positive first. And also, this group had a slower rate of amyloid PET accumulation. So the conclusion from this paper was that we added information, we added that fluid and brain markers give us complementary information. And then after that, that paper was focused on dichotomous biomarkers, but then we continued, and this is a paper that is going to be published, is already accepted, and is not published yet, but we propose a continuous model of amyloid biomarker discordance in the same set from ADNI, and then we validated it in other AMIPAD cohorts. We developed a continuous model where we fitted CSF and PET data to a hyperbolic formula, and we used the distance, Euclidean distance to the curve, as a measure of how much imbalance there is between PET and CSF biomarkers. So towards this side, these points have a greater PET over CSF positivity imbalance, and the opposite to the other side. And this, we call it amyloid aggregation score. This is the paper that will come soon. The first author is Sophie. These are people from Amsterdam, also Lidwin, and from Barcelona Beta. Okay, so I just wanted to show you that we have validated the data. Both we have a discovery cohort in ADNI, and also we have a validation cohort from the AMIPAD cohort. And yes, so what I just told you in the previous slide is this explaining the amyloid aggregation score. And we found that this imbalance, also we replicated the effect of the ApoE4 gene that I mentioned from the dichotomous paper before, and it can be seen more clearly here. So again, even using this continuous model for this aggregation score, we found that the group with lower aggregation scores had less ApoE44 carriers. But in summary, what we wanted to carry, the main message from this paper is that the imbalance between CSF and PET gives us information about heterogeneous pathways to the development of Alzheimer's disease.
And Elena,
yes,
you said in the slides, if I understand correctly, that becoming PET positive first in amyloid is correlated with a slower accumulation.
Yes, which sounds strange.
Yes, very strange.
But I know, but you are able to, maybe you can detect it, but you're not getting information on how fast. It may be that the plaques appear earlier, but not detected yet in the CSF, You can detect it. Yeah, it's true. It sounds like strange because you detect it earlier than you can measure it in the CSF, but it builds up slower in the brain. Yes. Because I know it's a bit counterintuitive, but this is what we observed. And actually, yeah. Yes, they were more likely to be CSF first. Twice as likely, yes.
CSF first.
Yes,
because that was, we previously thought that CSF it becomes positive before.
Yes.
But yeah.
I think that many other papers, the common knowledge is that amyloid is detected first in the CSF. And I think that other papers, because we review the literature,
common
I know.
Clinical knowledge.
Yes.
I mean, what we clinicians think is that CSF, but it is true that in the new proposal of criteria of the Alzheimer's Association, they say that it's not that it's not that clear, and so actually they don't use this concept.
Yes, but we also got many comments in conferences because the common knowledge is that always CSF comes earlier. But we found really that it's not always. I mean, still is more common. This pathway is more common. See, 7% goes this way, 3.5% goes this way. So it's less common, but it's there. It's a small effect. But yes, so what I wanted to say is that when we review the literature, we think that some other papers have excluded this group because it was too small. So maybe you need a bigger set like we took at that point, it was the bigger set and we were able to see it, and maybe other groups had smaller cohorts where it's kind of statistically you cannot find this group.
So you accept that the abnormality of amyloid in CSF appears before that in PET, that means that PET is less sensitive than the CSF measures.
Yes.
Because we accept
Yep.
the common knowledge, as you say, is that the lower values of amyloid in the CSF are due to the deposits.
Exactly. Yes.
So the conclusion is that PET amyloid is less sensitive than CSF.
For
this
that
is a clinical consideration.
For that group, yes, but for the other group, not. I think that maybe there are variations in the type of plaques, for example, that can be in part due to different genetic factors. But yeah, I don't know. Should I continue? We can come back later. Okay, so more recently, as I said, another biological mechanisms are important, and I found this paper quite interesting. The Hampel is a review on efforts to develop additional factors that are important in Alzheimer's disease, and they call it the ATXN classification, proposing that these X additional factors are the immune system, so inflammation, and even vascular factors. Yeah. So part of the research we did with Agneta was our interest. We are interested in studying glial cells. Neuroinflammation is, we find that it's a multi-stage process. It's quite complicated because it undergoes different phases with different markers. So I think that the challenge of studying inflammation is that it's not a simple biomarker, but there are many multiple biomarkers. Also, astrocytes and microglia undergo many different phenotypic changes. In some phases, they can become enlarged. In other phases, they become atrophic, and there is also glial cell death. So all of these changes can be reflected, for example, in microstructural changes in the brain. Yes, so I just wanted to introduce this, that to make the distinction that inflammation is intrinsically a very complex phenomenon. And then by also reviewing the literature, there is this idea that well, we don't know where inflammation is influencing the pathology. And I think it depends on which marker we are looking at. So there are some studies saying that inflammation is very early and that is caused by amyloid. And then that inflammation drives tau pathology. So perhaps inflammation is something that is in between amyloid and tau. But anyways, this is the topic is so complex, but I just wanted to introduce a slide to say that we are dealing with a complex problem. And finally, we also have biomarkers for N, neurodegeneration or neuronal injury. This is not specific to AD, but we have, of course, structural MRI, atrophy, cortical thickness. We also have diffusion-weighted imaging, and I've been focusing quite a bit on this in the last few years. And a very good marker is now CSF or plasma NFL, and even just like recently, neurofilament light, for example, has cleared is being recommended as a FTD biomarker for clinical trials. And then, so I have an interest in fluid biomarkers and especially plasma biomarkers because they are so cost-effective and they are non-invasive. So I've been interested in looking at, can we see inflammatory markers in the blood, and do they have prognostic value in MCI or in Alzheimer's disease? In this recent paper, they looked at multiple inflammatory markers, including cytokines, chemokines, and growth factors. These are panels of markers measured with Olink panel. And in this paper, we read that these multiple plasma biomarkers of inflammation are predicting the cognitive decline in patients with mild cognitive impairment. So we were motivated by some of the readings in the literature on this topic. So during the last year, I've been supervising a PhD student. She's Mona Lisa Malart. She has just graduated in January with her PhD. So the last paper of her PhD, we wanted to investigate what are the associations between amyloid, tau, and peripheral inflammation, as well as episodic memory in older adults. So as you know, all of this data, if we had to acquire it ourselves, is very expensive. No? It's difficult to access, but there is more and more data that can be requested. So we were searching online in NIH projects, and we found from Columbia University that they recruit Hispanic populations from Manhattan. And this is also a very interesting topic now in the field, because one of the interest is replicating biomarkers in different populations, in different types of ethnicities. So we found that as a cohort that had a very rich data set, including T1 weighted MRI, also flare MRI, amyloid PET, tau PET, and the same oleic inflammation panels with 92 proteins. All of them have to do with cytokines and inflammatory factors in the plasma. Out of these 92, we had to some of them were very much below the limit of detection, so we picked only the 76 that are above this limit of detection. And so we obtain a cohort of 132. It's called late middle age Hispanic adult. So on average, they're about 60 years of age, 64 years of age. And we had the idea so out of these 132, we decided to split the sample in equal groups based on median split of amyloid and neurofilament light. This way we get 4 groups. So this group is amyloid negative, NFL negative, amyloid positive, NFL negative, and amyloid positive, NFL positive, and a last group, which is low in amyloid, but high in NFL. Okay, so they had similar age, similar, I don't remember if statistically there were differences in how many women and men in each group. On average, they have between 10 and 11 years of education. And the group with highest proportion of ApoE4 carriers was the amyloid high, NFL high. Okay, so when we split, so these are the groups, we call them 0, 1, 2, and 3. So 0 has low plasma NFL and low amyloid, and then group 1 has high amyloid, low NFL. Group 2 has high NFL, high amyloid, and group 3, high NFL and low amyloid. So the first thing we wanted to see is, do they already have elevated tau? Which groups have elevated tau measured by this Merck 6240? And we found that it was only the group with both high NFL and amyloid. Sorry, I say it again. The group with already high amyloid but low NFL had high tau in middle inferior temporal cortex. And when you already have plasma NFL is high and high amyloid, then you have high tau both in the middle inferior temporal cortex and in the entorhinal cortex. Okay, so this, yeah, internal tau. Yes. Yes. Compared groups. Exactly. I forgot to say that. So everything we are going to compare with the 0, 0. So with the group that has low amyloid and low NFL. And I want to also say that this high and low, we had to do a median split because these are quite young individuals. So if we apply amyloid cutoff, that typical amyloid cutoff, only less than 10% of people are amyloid positive. So this is one of the, I don't know if it's a limitation of the study, but we don't pretend that we are having let's say, a clinically valid amyloid cutoff. But it's just a way for us to try to make sense of the data, right? Yes. because I think we think that this middle-aged group is a very interesting group to look at very, very early changes. Okay, so after looking at the amount of tau, we then look at cortical thickness using the typical AD signature cortical thickness. So we were expecting that perhaps the amyloid positive, NFL positive would have already some thinning, but actually we found that only the group that has high NFL and low amyloid was the one that has some thinning, right? I mean, I would expect that also this group would have thinning, right? Because it's related. But interesting also, this group has high white matter hyperintensities, which is a marker of cerebrovascular injury. So, of course, we wonder, what is group 3? Is this a group that is on the pathway to AD, or are there maybe comorbidities, perhaps, right? Likely. And also, being as Hispanic population is from Manhattan, is different than other white populations that had been studied before. So, that, yes. Because
neurodegeneration should be less pronounced than in the other groups.
Yes, so you think that to be part, so group 3, you think is not on the pathway to AD?
No, because then neurodegeneration,
yes,
and also white matter hyperintensities that are well-known marker of advanced AD should be lower than in the other groups if the others had more advanced AD. I mean.
Yeah, I would expect that I would expect that the group 2 with high amyloid and high NFL would be the most with more cortical thinning as well. What do you think?
Yeah, I mean the most obvious interpretation would be that that this third group has some other neurodegenerative condition if there's no amyloid by definition. So I wonder if there's any evidence from the MRIs that the pattern of atrophy is different, because just because they have atrophy in the AD signature region doesn't mean they have an AD typical pattern, because they may have atrophy also in the occipital lobe or somewhere.
This is a very good point. And I think we should also not just to take the AD signature cortical thickness, but maybe other more neutral thinning, right? Or looking also, like you said.
Or at the whole brain level.
The whole brain, yes. And we are submitting this manuscript. This might be a question from reviewers to ask to check other.
You can suggest Mitchell.
So I mean, because we see that, but we see that a lot AD signature regions. But if you don't check for other regions, then you may have just atrophy anywhere, and that doesn't make it an AD pattern. You know.
And this is also one of our co-authors is Konstantinos Kiotis, and he quickly said, just because it's an AD signature doesn't mean that, because it may have also atrophy in the frontal lobes, and we haven't checked. So, but I think,
not certainly, but very likely a different neurodegenerative disease, because white matter hyperintensities is a characteristic of advanced AD.
Yes. Yes, but it's a good point. It could be other neurodegenerative diseases, because, like you said, you would find it advanced AD, and these are definitely not advanced, because cognitively they are normal. Some of them may be on the preclinical AD, but definitely not advanced, because no one was objectively impaired. They were all cognitively normal. Okay, and then the last, so we checked these two things, but then the last column was plasma inflammation, because I think, and this is a subject that has not been looked at so much yet, I think. And so we went to look at if there are, so the box plot of inflammation is missing from here, but I can tell you that it was high only for group 2, so when they have both amyloid and NFL, the AD group, but also the group 3, the one that has high NFL and low amyloid. And actually, we did multivariable linear regression models to try to understand what are NFL correlated, what are plasma inflammation correlated with. It's not correlated with amyloid, if we do a model, not with tau. The only thing that plasma inflammation was clearly correlated with is NFL. So NFL is a marker of neuronal injury. So at least in this cohort, we see a close correlation between having high plasma inflammation and neuronal injury. We don't know fully what it means, but we didn't see a connection with amyloid or tau in this cohort. Maybe it's because it's too early. Maybe to see connection between plasma inflammation and amyloid and tau, we need to look at MCI, for example. No? But I mean, this was the cohort we had. And also interesting, plasma inflammation was related to high body mass index and diabetes. So we think that also there are comorbidities here in this group. So the group with low amyloid and high NFL, they might not be on the path to AD or FTD. Maybe they have these comorbidities. I don't know. What do you think? The comorbidities can cause injury in the brain, but so this is comorbidity. Yes. But and then the last aspect that we are looking at is cognitive measures. And again, that was also a little unexpected. We found that this group 3 that has high NFL is the one that has low episodic memory. And we didn't find that effect with the group with high tau. We did not see a correlation between tau and episodic memory in this cohort. So this group also has cognitive impairment. Yes. So finally, I have to say that all these models were corrected for multiple comparisons because we have 76 plasma inflammatory markers. So we always corrected for 76. So we believe it's quite strict, the analysis we did. Yes. So we need to explore more this topic. But yeah. Any ideas?
No ideas, but another question.
Okay.
So you mentioned that you found this data set looking for NIH-funded projects, which are like obliged to share their data, right?
Yes.
But all of these variables are actually also available in ETNI, for example.
No, these ones I couldn't find in
Plasma NFL is available, amyloid PET,
MK?
tau PET.
And
well,
plasma
not
inflammation?
MK, but like fluortaucipir.
Okay, but plasma inflammation I couldn't find.
There are certainly some plasma biomarkers there. I'm not sure if for all of the individuals or for the, but at least like for the first part of this presentation.
Sure.
Like
Yes. No, I think so. I think the reason we searched for this data is that Mona Lisa's thesis was the 3 first papers were all in vitro studies with second generation tau PET. So we wanted for her to get experience with second generation tau. So we started to look and we couldn't find second generation tau PET in AFNI with inflammation. I mean, I couldn't find. I don't know if there is now. But. So and then we found in this cohort that originally the cohort is to explore diabetes and risk factors and body mass index in Hispanic populations. Okay. The connections between diabetes and Alzheimer's disease. And they made the data available. And we searched their publications and they had not done this analysis. So that's why we, but maybe in the future we could try to replicate it in other cohorts.
Yeah, I don't think the difference between MK and fluortaucipir should make a big difference. According to Bill Javis, according to Bill Javis, the main difference between second generation tracers and fluortaucipir, which is the first generation tracers, is that the others were late to the party. That's his
late to the party. But I also heard that second generation tau PET tracers might be sensitive, more sensitive, especially for cohorts like not so old people and cognitively normal. I don't know. Could be, but you're not sure. Okay. So have you looked at? Yeah, I'm very curious that if, because when you look at pictures of fluortaucipir and MK, it seems like to me even visually, fluortaucipir has more non-specific binding.
Yes, that's the main point of developing the others.
That's why I think that having a tracer that has less non-specific binding will allow you to pick up small accumulation of amyloid.
The non-specific binding is usually very specific to some regions.
Right.
So
it's not going to.
Basal ganglia, especially.
True.
But it's true that some of the next generation tracers have a larger dynamic range.
Yes, that's
this
true.
This effect has often been interpreted as providing more sensitivity to.
Right.
Signal, but
yeah.
I haven't seen like
a strong. maybe head-to-head studies could be interesting too.
Another thing that I find interesting in this study is that it seems from intuition, very basic intuition, that group 2 should be more advanced than group 1, right?
I agree.
But
and it's not.
Propagation of tau in your regions seems to be contrary to what we might expect from.
Although I think it's also the
drug
relatively,
staging.
I think the relatively small sample makes it that these are, small variations.
See, now it makes sense to me. I mean, what you're finding here is individuals that are clinically affected with probably a vascular dementia. So that you're probably this cohort is enriched in these individuals because they are looking for that. And you are detecting the group 3 probably because they are clinically affected, so they are more advanced. So it makes sense.
But they're still cognitively normal.
Yeah. But when you do testing, they are more advanced than the other that have.
Yes, and to us it was interesting that this cohort also has a high body mass index, which was associated with plasma inflammation. And then we found one other paper with the same O-link panel, found also BMI positively associated with plasma inflammation.
The other thing I wanted to say is that you can replicate this in our core because we have O-link for almost 200 individuals.
You have O-link?
Yeah. In Vallecas, the 3K panels.
It would be super interesting.
And we do have, we do not have tau PET, but we do have, of course, NFL, and we have amyloid.
Or you have phospho-tau.
And we have phospho-tau, and we may have MTBR tau, which is the closest thing in biochemical markers that we have to PET-tau soon.
Yeah, and do you collect also like clinical information?
We do have,
if they have
course,
diabetes? Because it could be really, we could replicate it, maybe not with the tau PET, but with phospho-tau.
We do have this information, yes.
Yeah. Yes. So, this was just one study, but it made us think a little bit for the future. What are the implications from this study? We think that, but this is a little bit like kind of thinking for future studies, that developing Alzheimer's disease pathology can be via different pathways. In one, you have AD pathology specific with high amyloid and tau that may lead to inflammation and neuronal injury later on. But we think that perhaps we can have comorbidity driven with chronic inflammation leading to neuronal injury, and that this may enhance the risk of developing AD clinically. There are some epidemiological studies showing that, and there may be mechanisms involved, and it could be the inflammation, chronic inflammation, have an impact on the brain. There are studies, but I'm not an expert on the biology going on between the periphery and the brain. And I think it's still quite an open research topic. I don't know what you think. And then the two-hit hypothesis. You have amyloid and tau, you have inflammation. One may lead to the other, but both of them together, they may synergistically contribute to enhancing or making AD develop clinically at an earlier age, for example. But for this, we would need bigger data sets. We need longitudinal data, of course. Anyways, I just wanted to show that this is kind of, I think, what biomarkers help us to understand the biology of the disease. And the last part of the presentation, I am okay with time, right? Or no? Okay. But just stop me if I talk too much. And yeah, so I know that you're also working on microstructural measures, so I thought that's why we also talked about this topic. We are very interested to also explore the impact of the pathology on the structure of the brain. And traditionally, we have T1 weighted MRI, but with diffusivity, we can now explore the gray matter. And as you know, DWI has been traditionally used to look at white matter tracts. But in collaboration with the Hospital San Pau, with the group of Juan Fortea, we applied a pipeline that they have developed with Victor Montal. And yes, as you know very well. And the idea is that even before you have clear thinning of the cortex, you start to have synaptic loss, some neuronal loss, and this may be reflected in a greater movement of water molecules in the gray matter that can be measured as increased cortical mean diffusivity. The pipeline is actually available in how do you call GitHub, so it has been shared, and there are other groups starting to apply this pipeline in different cohorts, but it uses tools from FSL to co-register T1 weighted MRI and DWI, and the important part is using FreeSurfer to perform analysis on the cortical surface. So yeah, as I said, we use FSL5. The pipeline incorporates FSL5 to do the co-registration, and then the data gets projected on the surface, so we can compare on a vertex-wise or even regions of interest. Yes, we can extract for each region of interest the average value of cortical mean diffusivity for each region. Yes, so this is the co-registration between DWI and T1 weighted MRI, and it uses BB register. Yeah, and then finally, so we have full whole brain cortical mean diffusivity maps right now for every subject, so we can do vertex-wise analysis, and we always extract in the Desikan-Kiliani atlas. So we have 68 regions of interest. We have originally, the first study that I was involved with is in collaboration with the Harvard Aging Brain study. And the hypothesis, well, the interest of the study was that we look for biomarkers that can predict short-term cognitive decline that can be used for clinical trials. So we want biomarkers that predict cognitive decline over 2 or 3 years. And as you know, amyloid pathology predicts cognitive decline, but over a long term, maybe 10 years, 5-10 years. And if you add complementary non-invasive imaging biomarkers, we might be able to identify individuals at risk of short-term decline. So as I mentioned, we were investigating cortical mean diffusivity in the gray matter. Okay, so I'll just go directly. So we had 196 participants from the Harvard Aging Brain. We split the sample into those that are amyloid positive and amyloid negative. About 25% of the people are amyloid positive. And all the individuals had floor tau CPIR, tau PET, DWI, amyloid PET, and all the cognitive measures, as well as a subset had multiple longitudinal MRI, cognitive, and clinical follow-ups. The first question we had is whether the mean diffusivity is more associated with amyloid or tau. When we did the correlation between whole brain cortical mean diffusivity and amyloid, we found no association in this cohort, but it was associated with tau burden, and these are the significant regions of interest after correction for multiple comparisons, illustrated here in the inferior temporal FTP, floor tau CPIR region. We built mixed effect model with random intercept and slope to extract the individual random slope of cognitive decline. This random slope of cognitive decline we use as an outcome variable in different, these are the models, multivariable general linear models now. So we want to see which set of biomarkers best predict the rate of change of episodic memory decline with a PAC5. So the main finding from the study is that cortical mean diffusivity, when it's underlined, means that it was significant predicting the memory decline. And so it did predict cognitive decline, and interestingly also after adding cortical thickness, so as one of the predictors. So the main message from this study is that cortical mean diffusivity is able to predict faster cognitive decline with more sensitivity than cortical thickness. So we think that mean diffusivity adds value. It adds more information than just given by the cortical thickness. Perhaps you are also having this hypothesis in your cohort. We also found, using similar models, that the baseline levels of cortical mean diffusivity can predict the future rate of hippocampal volume loss. There was no significant interaction of amyloid or tau with cortical mean diffusivity at baseline in predicting the longitudinal rate of hippocampal volume loss. A few percentage of the individuals converted progressed from cognitively normal to MCI, and the group that had high cortical mean diffusivity significantly predicted progression to MCI. Actually, well, we did some post-hoc analysis, but also tau PET at baseline is able to predict, as many other studies have shown, this is a very strong predictor of future cognitive decline and also of progression to MCI. And in a more recent paper, a follow-up from this study, we also found that cortical microstructure at baseline, so cortical mean diffusivity, predicts longitudinal accumulation in those that are amyloid positive. And the effect was quite small, as you can see. More data is needed to validate these findings. And then, yeah. Yes.
Even we can see them even earlier than we see tau accumulation.
Yes.
And
or it can be coexisting. I think it, yeah, exactly. The problem I see in many of these biomarker studies is that we have a problem of multicollinearity. Because if you start to look, almost everything correlates with everything. No? For example, and just as a follow-up, there are this group from Lund, Nicolas Potorno, that is focusing also on, you know, these studies. He found that cortical mean diffusivity is correlated with amyloid. It's also correlated with GFAP, and it's also correlated with tau. It also predicts future accumulation of tau. So I think that, I think many of these variables are coexisting. So tau has an impact because it leads to synaptic injury. That also increases the mean diffusivity. That also leads to more tau accumulation. So, but maybe it's difficult to clean out what exactly causes what.
So regarding tau, it has been seen in the neuropathological studies that the tau PET can only measure advanced tau pathology.
True.
So maybe tau is already there. So
yes.
Mean is not predicting tau, just it
yes.
Yet to measure.
Yes. Yes. That is a very good point. Maybe, but this also gives value to our cortical microstructure measure, because maybe it's very sensitive and it's much cheaper than tau PET. Thinking of tools that can be used in the clinic. And I think, yeah, of course, you don't have the biological signal. It's a little more difficult to interpret. But maybe correlating it with fluid biomarkers, you can combine MRI, DWI, and fluid biomarkers and try to understand what are we measuring. Yes, so again, studies from this group from Lund also suggest that compared to cortical thickness, mean diffusivity is reflecting some other processes in the brain with more sensitivity. This is the most recent. I haven't found many longitudinal papers. I think this is one of, maybe the first one. It was published this year. And they have found correlations between changes in mean diffusivity and changes in GFAP, as well as changes in YKL40 and in plasma NFL. So, and the patterns are quite similar. Seems quite strong with GFAP, so this suggests that perhaps it's an early sensitive biomarker of damage. Do you have any question, or you have the answer? Well,
we have studied GFAP in our clinical pathological cohort, and we found that it was the serum levels of GFAP were correlated with the activation of the astrocytes. We really were able to correlate with the histopathological changes, with the area of quantifying the area of GFAP staining, so kind of a proxy of the activation of the astrocytes, so they are bigger astrocytes, and we correlate this with the serum levels. So
you think
I don't know. I don't know, but what is the biological substrate of this mean diffusivity? But gliosis, astrogliosis, definitely could be a candidate.
Could be a candidate. Yes. Okay. So since I work in I wanted to show you a couple of examples from data that we have at Karolinska, and one of them is the Swedish familial Alzheimer's disease study. And these are data collected from the 90s in a longitudinal design. It's not a very large cohort because, of course, these are rare families. There are 6 families. In total, 81 individuals with 219 visits. Last year, we published for the first time the plasma with CMOA markers. We characterized the cohort. So we have GFAP, NFL, p-tau 181, and total tau. And these were the results published. And I think that the most clear is this scatter plots with respect to the estimated years of onset, where we reported that GFAP is the one that changes earlier at about 10 years before onset, followed by phospho-tau 181. And then NFL changes at around the clinical onset, and there was no difference in total tau. Total tau seems not to be a very useful biomarker because it doesn't differentiate carriers versus non-carriers. And then we have one study on mean diffusivity in the same cohort. Again, it's a even smaller data sample because these have to have PET and also MRI and DWI. But we found this pattern where in red you have mutation carriers, and mean diffusivity goes from low values at pre-symptomatic stages and higher values at symptomatic stages. This was a pilot study, it's quite small. But we don't have more familial AD data. But yes, and also in this year, we put together the GFAP, the fluid biomarkers with all the PETs. And this is the summary that, if you want to know more about it, is published in Molecular Neurodegeneration. And it's combining all the different PETs and fluid biomarkers, where you can see the GFAP about 10 years before onset, and amyloid is the earliest, while FDG declines with years of onset, and deprenyl, this was the marker of astrocytosis, is highest early on and declines with the progression. And then finally, as I was talking before, I have in the last couple of years, I'm focusing on FTD, and with Caroline Graf Group, she's the head of the Swedish FTD.
Lena,
yes.
Just to come back to the previous, this is very interesting results, right? How deprenyl goes in a
completely
different direction to GFAP, right?
Yeah, I know.
It's telling us how complicated this.
Totally agree. And we many times wonder about this result. We had comments at conferences whether the kinetic model was wrong, because perhaps some people say that you're using the cerebellum as a region of interest, but maybe you have astrocytosis in the cerebellum, or I mean, there can be some problems. But we replicated in this paper, the first author is Konstantinos Kiotis, and he repeated all the kinetic model with a better model, and he still gets the decline of the prenyl. So we don't think, I mean, we cannot do more with this data. It's also a limited data sample, but it has been reanalyzed. So I was happy when he reanalyzed that he got the same, because I was involved in the first models, but yes. And perhaps, like you say, deprenyl measures a subset of astrocyte, a type of activation, who knows? And then GFAP measures something else. But yeah. And regarding frontotemporal dementia, this is a more rare the type of dementia. So we collaborate. So Caroline Graf, I work with Caroline Graf, and she is the head of the Swedish FTD initiative. But we collaborate with GEMFI, which is 35 centers, or even more now. And it's an international effort. And there are other efforts for genetic FTD, like the one in the US. And this is a paper that put together information from different biomarker studies. In the field of FTD, there are no, as you know, there is no good protein biomarkers yet. There is no PET tracer. They are not specific. It's not possible to image FTD. The 4R, 3R is also not good enough. So this is not available yet, but so that's why the most widely available biomarker is neurofilament light, and the evidence is that it starts to increase about getting onset of the disease. But also it's interesting that inflammation has been measured with different fluid biomarkers. And according to this study, the summary paper, it seems that is after the neurofilaments. But of course, I think this needs to be studied better. We have a lot of data from this cohort thanks to this multi-center study. This has been published. Is the atrophy patterns are very different depending on the mutation type, progranulin, C9orf, or MAPT. So again, we thought it would be very interesting to test whether cortical microstructure would be a more sensitive biomarker also in genetic FTD. There is a paper these are sporadic cases, so not mutation carriers, but it's also from Barcelona, using the same pipeline as Juan Fortea's group, and they found that on the top panel is cortical thickness and below is mean diffusivity. So there is some evidence that mean diffusivity is more widespread, has a greater effect size when comparing patients versus controls. So we hypothesize that we will be able to capture early neuronal injury better with cortical microstructure also in genetic cases of FTD. We think that perhaps cortical microstructure has a potential to track early neuronal injury induced by the spreading of tau and TDP43, and to track early neuronal injury with greater sensitivity compared to cortical thickness. And that also it will be more associated with cognitive dysfunction and predicting future cognitive decline and clinical progression. These are ongoing studies. Yes, so the aims, as I said, we want to investigate the whole brain surface-based maps and compare microstructure and microstructure, macro and micro. These are some ongoing studies from the group using plasma biomarkers. These are multiplex sets, and a panel of CSF proteins has able to separate genetic frontotemporal dementia from presymptomatic mutation carriers. And these are the 4 more strong effects. We found markers of neuronal injury. And in our case, we don't have neurofilament light with this type of multiplexes technique, but we have neurofilament medium, which is highly correlated with neurofilament light. A biomarker that comes up a lot in FTD is neuronal pentraxin 2. There is also the growth factor, BGF, and a marker of astrocytosis, aquaporin 4. In the next steps, we want to correlate these biomarkers with cortical thickness and later on with mean diffusivity. Cortical thickness in our cohort, these are also ongoing studies with a PhD student from the group. We replicate, this has been published before, the patterns that are different in C9orf, in cortical thinning in symptomatic carriers versus non-carriers is very widespread, while progranulin affects smaller areas and mapped mostly the temporal lobe and frontal lobe, but in much less effect size. And we are investigating all the associations with the CSF biomarkers. The main finding from these preliminary studies is that the marker that is common to all the mutation types is neurofilament medium, to be expected because it's a marker of neuronal injury. And then we need to go more in detail, but different markers come up only when we look at C9orf, for example, the aquaporin 4, and they don't show up with correlating in the other mutation types. So one important aspect of genetic FTD is that it's very much dependent on the mutation type. We are doing the analysis now comparing cortical thickness and cortical mean diffusivity. This is a subset, a very small subset with 153 individuals in total, but we have processed now already 803 individuals, and we are analyzing this data. But we already, in this smaller set, we see evidence that the effect size of mean diffusivity is bigger than cortical thickness and more widespread in the brain. So again, replicating a bit the findings in behavioral variant and in the sporadic cases reported in Barcelona. When we look at the cortical thickness versus age in carriers in red versus non-carriers in green, this is only a representative region. For example, the caudal anterior cingulate seems that mean diffusivity is able to find a greater effect at an earlier age before onset, which is promising because maybe it's more sensitive than cortical thickness, but this is really ongoing and very preliminary. So we are still processing and analyzing the data. And finally, we also have longitudinal data points, like 1,800 in total follow-ups. So we want to investigate whether this is only a hypothesis that the longitudinal rate of increase in cortical mean diffusivity will increase from non-carriers to presymptomatic carriers and to symptomatic carriers. From GEMFI, we have access to GFAP data and NFL, so we want to explore the longitudinal rates of increase in these plasma biomarkers versus the mean diffusivity. And yeah, so just a summary slide, these are the biomarkers that we have been looked at, and our aim is to try to understand the timeline of changes. And as we discussed before, many of these changes are in parallel and there is a lot of collinearity. So this was my last summary slide. And these are our collaborators. And these are, we get funding mostly from Sweden and some European funding. So thank you very much.
Very much.
Okay.
A lot of interesting stuff. More questions?
There are some questions there.
You had the opportunity to look on all this stuff with FDG-PET, because FDG-PET might be expected to be a very early prospect of microstructural changes also.
Yeah.
Similar to.
But do you have, so I have the feeling usually that FDG is not too early, no? It's usually
earlier than
cortical thickness. That's true. That I don't know. I never saw any study, but it's true. I don't think I have seen any study comparing cortical mean diffusivity and FDG-PET. Maybe you have the data.
One of the things that we could do to try to clarify what is behind this mean diffusivity phenomena is because we have in our clinical pathological court, we even have post-mortem MRI, and then we have the neuropathology, and we are quantifying.
Yes, this is a very good. Yes,
we do have the sequence is not that good, right?
Yep.
Could we do that? I mean, we not only the post-mortem MRI, but
but also in vivo MRI.
We have in vivo MRI as
yes, I think yes, we haven't tried that yet, but also in Karolinska. So the Karolinska is the head of the brain bank there, and we collect as much as possible whenever they donate. There is a collection as well, but we haven't looked at this point. And usually they have a protocol when the patient donates the brain, they get quantified the amount of tau in different regions following an in-house protocol. And also they look at TDP43, and there is a paper that's showing type A, B, C, D, E. I don't know if you also look at this. Well, I don't know if it's, different types of FTD, and we have some. Yes, so we have this data as well. Do you look at the types or?
No, we just look at TDP43.
TDP43.
It's not here.
Yeah. Okay. Yes. Yeah, so I think I totally agree that since until we have PET data, it's very interesting to look at post-mortem, antemortem, post-mortem studies.
We should look at a logical question.
Yes.
It seems you have worked with quite a lot of different cohorts, so different types of diffusion-weighted imaging acquisitions. Do you have, including in the same GenFi cohort,
I know.
you have different acquisitions, and then the one from Barcelona, your group?
I know.
Do you have any feeling of how the number of directions or the B0 field may affect these diffusivity measurements?
Yeah, that's a good point. And first, actually, so Victor Montal, I has got this question from the 2018 paper, cortical microstructure in healthy individuals from Spain from multiple centers. If there was an impact of how many directions. And in the reply to reviewers, they have done a very careful study simulating data, removing directions and checking, and he proved it that there is no big impact. So he has shown it in the it's not published, but he had done the exercise. because I asked, we have very different amount. Some people have 12 directions, some have 64. So I agree, but he has shown that it's not a strong impact. Then we use
with some methodological studies showing a similar effect on like on diffusivity measurements, but these like traditionally all of these studies have been focused on white matter tract integrity, FA of white matter pathways. So and the general conclusion is you really just need the tensor because these are diffusion tensor imaging metrics. And for a tensor, you just need 6 directions. And if you want to have a more robust assessment of the tensor, then 12 should be fine. And the higher directions are more for robustness and for track if you want to do fiber tracking. Actually.
Yes, that's a very good.
For these diffusivity scalars,
apparently it's not so.
I wasn't aware of any study showing a similar like comparison for cortical MD measurements.
Yes.
Much more.
Yes.
Novel or less used.
Right. But I can put you in contact with Victor because he has the reply to the reviewers and he has all these studies done. Yeah.
That's very interesting.
Yes, and also we use gemficide as a fixed factor in the statistical model to try to correct, but I know it's kind of very rough way. And we are also applying combat. You know, this combat, many groups is very popular now. So we are doing all the analysis without combat and with combat. I think we compare box plots and combat is doing something to harmonize more. So I want to be able to, when we finish the analysis, to see if there are differences or if the site is really influencing the results or not. Do you use combat as well, or when you harmonize across centers?
So common to use
combat
in the context of APNI because
that's true. In APNI, people don't use really. You don't take into account differences.
They did a good job and good PR for having harmonized the coefficients. I don't know if that's enough actually, but they did all the PR.
Maybe they do, but I don't believe that the data is so harmonized because they get data from everywhere with all kinds of small and big.
It's not a post acquisition harmonization.
Yeah.
So it's just there is an ADNI protocol and you need to validate it with phantoms and everything before pre-acquisition.
True.
Still there are differences between centers, but it's you are in a much better situation than the common.
Yes,
that's right. I forgot about especially the DTI sequences and even ASL. Like these kinds of sequences are only acquired in some centers who all have the same scanner. For example, I think DTI is only acquired on Siemens scanners within the APNI context.
Okay. And I know that GEMFI has many different Siemens and GE and Philips, so it's very diverse.
APNI is very strict. It's not perfect, but there is an extra effort to ensure a priori
the
scans are as similar as possible on acquisition.
Okay, on acquisition.
Yeah. Questions? Any last question? Because it's like
it's already
5. Yes. So we can all give an applause to Elena for that great presentation.
Thank you. Thank you.
Another applause because he came from Stockholm just to give
for Thank you. Gracias. Tenemos mucho